Your robot worksheet, I'm going to show you how to fill it out. You're all gonna get a different named robot, and all of the robot's genes are lettered. So you have gene A, B, C, D, E, F, and G. So the first thing that you're gonna do is you're gonna copy over gene A over here on this side of the worksheet where you also have it stating gene A. So I'm just gonna go ahead and do it just like it has on the worksheet. It has every 3 base pairs together. So I'm gonna fill it out just the same. Here I have AAG, then I have GGT, TTC, and CAT. This is a gene. This is a segment of DNA that codes for a protein. So I need to decode it and make the mRNA. So now I gotta go in and do transcription. So remember, A will make a U because there's no T's in RNA. So here I have U, U, C, and I'm gonna keep going, and now I'm making codons, because remember, every 3 bases in mRNA is a codon. GGT would transcribe into CCA, then TTC would transcribe into AAG, and CAT transcribes to GUA. Next, where it says AA, this is amino acid. Now you gotta break out your handy dandy amino acid table, and you need to find the right amino acid based on the codon. So first I have UUC, UUC. This is phenylalanine, so I'm gonna put my phenylalanine. CCA gives me CCA, proline, and this should go pretty quickly, you guys should know this. AAG, AAG, lysine, and then GUA, which would give me valine. Okay, this is my one gene A. If you want, so it doesn't confuse you, you can go ahead and separate all the genes with a marker or a highlighter so they make sense to you. Now I gotta find out, well, which trait is my robot expressing with this gene? On Schoology, you guys will see this robot student reference sheet that's gonna have all of the amino acid sequences there for you. So I'm gonna find mine. My robot had phenylalanine, proline, lysine, and valine. Now I need to find it and match it. I can see it right here. Phenylalanine, proline, lysine, and valine. This trait tells me which antenna my robot has. So my robot specifically has a single antenna. So in this little box, I would first draw it so I don't have to go back later and find out what that looks like, and then fill out single antenna. Then you do that for the rest of the genes all the way through. But one thing you're gonna notice is down here, it's talking about a mutation. Here, back to the lettered genes, you have a mutation. My specific robot tells me I have a mutation in gene A. So here it tells me my original gene A, and here it tells me my mutated gene A. So what we're gonna do here is I'm gonna write down the mutated gene. So now it's AAT, and it goes to GGT, and then I have TTC and CAT. Then you're gonna do the transcription and the translation. You will get a different set of amino acids here than you did originally up here in gene A, which means it's gonna give you a different trait. You're gonna notice from your original to your mutated DNA, if you are missing a base, you're gonna need to shift everything over left or right to keep all the bases in a triplet of 3. Okay? All the bases need to be in a triplet of 3. If whatever mutation yours gave you didn't let you fill up a complete triplet, that's okay. It's a mutation, something went wrong. It's going to affect your trait somehow. Once you've done that, on the back of your worksheet, you're going to have a place to draw what your normal robot looks like and a place to draw where your mutated robot looks like, okay? So you're gonna have differences because there's a mutation. Down here where it says normal gene, this is where you rewrite your normal gene. Here I have my mutated gene. Now if you remember back here, my normal and mutated gene was all gene A. So I'm gonna rewrite the DNA again. So normally it's supposed to be AAG, and I'm gonna go ahead and write that. AAG, and then I have GGT. So I'm gonna fill in GGT. This is still normal. And then TTC, TTC, and then lastly I have CAT, CAT. Over here where it says mutated gene, I'm gonna now write the mutated version of it. Now I have AAT, AAT. I can already see where the mistake is. GGT, and then TTC, CAT. Then, based on what the differences that you see in your normal and in your mutated gene, you're gonna tell me which type of mutation is that. If you have the notes already, then you can tell me specifically, is this insertion, deletion, substitution silent, or substitution missense? If you are missing those notes, don't worry about that part yet. We will do it together next time. Okay, so this is how you fill out your robot worksheet. Transcribe and translate all the genes. You will have this reference sheet on Schoology for you to access. Some of the amino acid sequences, for some reason, don't match the gene in order. So let's say you're looking in arms and you can't find your sequence in the arms when it should be, look everywhere else and you'll probably find it. One thing to pay attention to up here though is it tells you, if you get the sequence, histidine, isoleucine, alanine, and stop, that tells you you have no trait. So if you were to get this sequence for antenna, this tells you your robot has no antenna. Okay, so don't forget about this one up here, and that's how you do the mutation worksheet.